So I presented in a symposium earlier today discussing lots of the novel oral agents in myeloma, but particularly focusing on the novel CELMoDs and how those activate the immune system or interact with the immune system. There's lots of really interesting data around this. We know that the IMiDs can do this to some extent, and the CELMoDs, being more potent Cereblon engagers, can probably do this in an even more enhanced way. So we can see data that shows that the CELMoDs can activate T cells, even T cells that have been exhausted, for example, after the use of previous T cell engager therapy. And really, the data suggests that these agents can lead to increased proliferation of T cells and also activation of NK cells, which really enhances their response to myeloma. So they have direct anti-myeloma activity as well, but by enhancing and engaging the immune system into that response, that can really increase their efficacy. And we're seeing that coming through in some of the early phase clinical trial data that Professor Richardson presented within the same session. I think what was really interesting is that this really fits in with some of the other discussions that have been going on at the meeting about all of the different ways we're now able to engage the immune system to target myeloma. So we have had lots of discussions about different T cell engagers, about different CAR T cell therapy approaches, thinking about all of the different agents, different targets, and how to sequence some of these agents either in combinations or before or after each other. I've just been in an interesting session thinking about how to manage some of the side effects that we're seeing as we have new agents come through, and we're then seeing more data around how to try and prevent some of those side effects in these agents that are new to us in the clinic.